Welcome to Lesson 8 of Module 3, Biological Diversity. In this video, we're going to be looking at the evidence which is used to support the theory of evolution by natural selection. So we've looked at what evolution by natural selection is. We've looked at the different types and how it might occur. In this time, we're looking at, well, what is the evidence? Why should we believe what Darwin and Wallace are proposing? So the evidence can be broken down into 5 main categories. So we've got paleontology, biochemical evidence, comparative anatomy, comparative embryology, and biogeography. The first is biochemical evidence. So we can use comparative genomics, where we study the genome sequences of different species and we look for similarities and differences. So the development of specific technology has enabled researchers to construct these detailed genetic maps. It has allowed them to investigate the role of each gene, but it also allows researchers to estimate when two organisms diverged based on the similarities and the differences in their genomic sequences. We can also have amino acid sequencing, and these occur with some proteins, so the sequence of amino acids that are found in a range of organisms. So, two examples are cytochrome C or hemoglobin. So the similarities imply that the organisms may have shared a common ancestry, but some differences imply that the organisms have evolved. And the number of differences is usually proportional to the length of the time since these organisms separated. Knowing this information allows scientists to construct evolutionary trees. Another one is DNA to DNA hybridization. So I've got the process here. So we can get the two strands of DNA, and these two strands, one is from one organism and one is from another, which are believed to be similar. And these are separated using heat. These single strands that are now formed are mixed with the single strands from another species. The two different strands will join to form a hybrid molecule. However, some of the pairs of bases won't match, and this is because they are different sequences. So the degree of this pairing and the degree in which they join together, also known as annealing, depends on the similarity of the sequences in the DNA. So a high degree of pairing or bonding means that the organisms are very similar, whereas a low degree of pairing means that they are very different. And the final one is DNA sequencing, and this is where we compare DNA sequences, and usually these sequences are similar gene sequences from different organisms. So organisms that have a common ancestor do in fact have fewer differences in their DNA. And this process is more advanced and reveals more detail. However, the techniques are really complex and expensive, and they do rely on highly specialized computer technology, as well as someone highly qualified to operate the technology itself. Here is a bit of an image about the DNA to DNA hybridization. We see the red and the blue DNA here. We can see that they're separated by heat and then allowed to join back together. And we can see that there's just one difference in that sequence of DNA strands. So we can say that these organisms are highly similar. The next is comparative anatomy, and this is where we look for similarities and differences in the structure of living organisms. So if they're more similar in their structure, implies they must have separated from a common ancestor more recently. Homologous structures are structures that have the same basic plan to their structure, but show different modifications because they're used in different ways. And the example which we have here is the pentadactyl, or the 5-digit limb. So we see here that all of these 5-digit limbs evolved from the lobe fin. And we can see here that they all have the same basic structure, but they're all slightly different depending on what the organism uses it for. So we can see it's quite similar. The bone structure is similar, but the bat has quite a different structure to that of the amphibian and to that of a whale. And they also perform different functions. Comparative embryology is where we compare the developmental stages of different species. Similarities in their development infers a relationship, and it's predicted that related species show similarities in their embryonic development. So we can see here an example of a few different organisms. And all of these organisms, as embryos, have the presence of the pharyngeal slits, which actually allow for the formation of gills. They all have tails, and they all have distinct muscle blocks. And that's found in fish amphibians, birds, and mammal species, all seen in this diagram here. But we see over time they then do evolve into different organisms, which don't always retain those structures which were originally in that embryo. Biogeography is the study of geographical distribution of organisms, and we're allowed to do that with both living and extinct organism. And Wallace's line is a suggested line, separates the distribution of animals and plants in Asia and Australia. So flightless birds, they're suggested to have originated from a common ancestor on Gondwana, and that the different populations evolved on the isolated southern continents as they drifted apart. So as a result, we have emus, ostriches, and kiwis and rheas, which all evolved from a common ancestor, but are now different because of the continents that they live on and which they were able to develop and live on. So we know that emus do look different to kiwis, but they have a similar, a common ancestor. And some organisms from Australia, so similarities to fossils found on other southern continents. And this is evidence that they may have had a common origin and later evolved due to the isolation of the continent. So we know that this is a form of divergent evolution. And paleontology. This is the study of fossils, and this provides direct evidence of the existence of an organism in the past. So fossils can either be mineralized remains in a rock or the actual remains of an organism, and it can be preserved in a range of materials. We do use index fossils, and these are organisms that live for a short time, but they're found over a wide area, and they allow us to correlate different layers of rock so that we can determine the relative age of rock. We can't actually find their actual age, but we can just look at them in relative ways. And we say that layers with similar index fossils are the same geological age. So here we just have a sequence of events of how a fossil does form. So we see that they're covered by sediments after they died, and we see that then we find them by erosion, and we can discover them later. But we can see that there are some fossils there remaining if they haven't worn away with that erosion. Transitional forms are we can find them in our fossils. We find evidence of them in our fossils, and these show the characteristics of two different groups. These do suggest that there's a possible evolutionary pathway of evolution where one group evolves from another. So one example is the crossopterian fish. And it has bones in its fin, which suggest it could walk on land. So we see that as an indication of where we evolved from fish into walking and land animals. And the second example is the Archaeopteryx, which is a small flying dinosaur with feathers. It is known to have appeared in the late Jurassic period. And so it shares features of both birds and reptiles. And this supports the theory that birds developed from reptiles. Relative dating is also used by paleontologists, and it states that the oldest layers are found at the bottom and the youngest layers are found at the top, unless there's been dynamic movement of the land on which we're looking at. So fossils in the relative dating methodology can only be described as younger or older than another, and we can do that by a few different techniques that are listed there. Then there's absolute dating, and this allows us to determine the actual age. We use radioactive elements that are present in the specimen. We do know that each isotope decays at a fixed known rate, and this is called its half-life. So the amount of radioactive materials left in a specimen will indicate its its actual age. And we know that this is an absolute age because radioactive decay is not affected by temperature or pressure or any thing in the chemistry of the environment. So one is thermoluminescence, and this is where we see the emission of light from a mineral when it's heated. So the amount of light is proportional to the amount of radiation. The older the object, the more light it emits. We can date calcium carbonate in limestone, coral, fossil teeth, and eggshells through electron spin resonance. And then radiometric dating is a form of absolute dating, which we determine the proportion of particular radioactive elements or isotopes within rocks around fossils or sometimes within a fossil. So depending on the radioactive element in which we use, we know its half-life, and we can use those things to date different types of fossils. So carbon-14, we can use it to date bones and teeth, and we can see that as we go down, there are different elements that have different half-lives, and we can see that they do allow us to find the absolute age of a range of different either rock types or organism remains. That concludes lesson 8. Make sure you tune in for lesson 9.